How do T cells attack non-self cells, and how is the major histocompatibility complex involved? All cells in a vertebrate express MHC class I protein, and the MHC class I protein has a function to present evidence of what's going on inside the cell. So fragments of molecules inside the cell, which could be self or bacterial or viral, get loaded into MHC class 1s, and then those get moved to the surface where they present the antigen. And a T cell receptor on a helper or killer T can match that, and if it does, can be activated through mechanisms I've talked about elsewhere. But another important piece to this is that the MHC complex 1 involves 20 genes, each with about 10 alleles. That makes for a huge number of combinations, billions of possible combinations. And the variation occurs in a specific location of that T cell receptor. The variation occurs in this binding pocket. So what that means is different MHC complex 1s have different shapes specifically in this binding region. Now all of them can bind peptides from inside. All of them have the capacity to hold small molecules from inside. But this particular region is different in all of them, and that matters for T cell activation. So if we have a particular cell that has the normal MHC1 for that cell, when a T cell receptor matches that, if it's a self, it's the normal self MHC1, then no T cell will be able to bind. And so with self MHC class 1s, the T cells are not activated, nothing happens. But if there's a foreign MHC class 1, in that case, there may be a T cell receptor that can bind. When that matching shape results in binding, that can then activate helper T cells and killer T cells. So if you activate a helper T cell and it releases cytokines to a killer T cell that also binds to that MHC1 from a non-self, maybe a transplanted cell, then you wind up with, here's our non-self and our fully activated killer T cells. And when they have a receptor that matches the shape, they release perforins that put holes in the non-self cell and reactive molecules, including reactive oxygens, and kill the non-self cells. And that's how transplants get rejected.